stood 50 in Munich. So I'm really honored and pleased to be here. Indeed, I have a mixed background, and after several years in academia and like 12 years in pharmaceutical industry, I joined the Radboud University Medical Center, which is one of the seven academic hospitals in the Netherlands. And basically, it's a fair site with an applied institute next door, the Max Planck. We got a university. It's about 11,000 employees. So it's a big bunch where we apply basically scientific insights, education into patient care. And that's the real driver after all those years to really make an impact on healthcare. And our mission is basically healthcare with care between brackets, because prevention is more and more on the agenda of these kind of academic hospitals. So our technologies are organized in so-called technology centers, and please check the link if you want to know more about that. But enough to say we got a good shop to do science. So when I'm teaching to my students, I'm frequently telling them that they're living in my future of medicine, because I was doing my PhD in the 90s, and it took me about 3 years to clone a gene. And now you can just order it online on Monday to arrive in your mailbox on Friday, which is totally different than the hard work we had to spend into cloning and sequencing genes at that time. Also, I was studying protein complexes, and it took me years and hundreds of Western blots to figure out which proteins are there. And now it's one day in a mass spec lab, and you know which proteins are there, but also the semic quantities, but also the modifications you have, which is just mind-blowing. So particularly as a molecular biologist, these developments in the omics technologies have been very, very stimulating. So it becomes really feasible to do a whole genome sequencing now in quite a short time at a reasonable budget to really apply it in research, but also in patient care, diagnostics, therapy monitoring, etc. But also the mass specs have been developed enormously to do the protein, glycan, lipid analysis, just to have 10 to 50 or 100,000 features in one assay. Real-time database searching is becoming possible. So you can really do while you do the analysis in like 10-minute gradients, you have the data at the end of your gradient, which is just opening new possibilities. So what I want to share with you is 2 examples, and I like to share stories. And if you want to know more about it, check the slide share where I put all my public lectures there. You may find that interesting. But I like to share 2 stories how we actually impact by applying these multi-omics technologies into personalized diagnosis and also personalized therapy, and then bridge it to the future of population health. But we see more and more that the omics technologies to analyze the building blocks of life, as we call it, are really enabling a diagnosis on a personalized level, opening options for personalized therapy. And together with the patient-doctor participation relationship, it's really the personalized healthcare model. And more these therapies become more expensive because they're more personalized and less market-driven, if you like. So the monitoring becomes more and more crucial, and the omics have great potential there. So I'll give you 2 cases. And one case is actually a few years ago when I was heading the large metabolic laboratory. I was approached by a mother from the northern part of the Netherlands, and she sent me an email. She said she wanted to visit our lab because we changed their lives. Obviously we were very curious. We tracked the names in our systems, but later they visited us and they told us this story. And the story was that about 20 years ago, they were a young couple with no genetic family history. So they called themselves normal Dutch people. They got a son called Brian, but immediately after birth, it turned out he had a very low birth weight, very acidic blood phenotype, low blood sugar. And soon thereafter, he developed movement disorders or movement disabilities, clearly had an intellectual disability, developed epileptic attacks. So they moved from specialist to regular care to specialist, different hospitals, could not figure out what it was. And at the age of 3.5, Brian died of a respiratory failure. Very black period in their life, very intense. They started blaming themselves. Did we do something wrong in the past or during the pregnancy? And there was no diagnosis. And I'll tell you in a minute why that was. So with still a child wish and no diagnosis, they thought, well, it may be just bad luck. That was literally their conclusion from the specialist. It could be bad luck. So they tried for another baby, got a son, called Joel in 2009, but he had the very same clinical phenotype. And in the end, had epileptic seizures every 1.5 minutes. And at the age of 1.5 years, he died from an epileptic attack. Now, obviously, this is a nightmare. Those among you who have children, well, I can imagine this is the worst thing that can happen. The doctor's suspicion was that there is a mitochondrial dysfunction. And several times during the day, we named mitochondrial. So as we are the only mitochondrial diagnostic lab in the Netherlands, we got those samples already early on. And what we get typically are blood samples and skin biopsy, so we can culture the fibroblasts to really study the mitochondria in the cells. And for those not fully aware, mitochondria are the energy organelles in the cell. They convert glucose into ATP, or just sugar into energy. And immediately the blood tests show that ATP indeed was very low. Other markers were low and high as expected with a mitochondrial phenotype. But when we tested the 5 mitochondrial complexes in those cells, turned out to be very normal. They functioned as they should, so we can test them functionally per complex. So we culture the cells and test these. At that time, we sequenced several candidate genes, the known suspicions, the known suspects, basically, but we couldn't find a variant. So we couldn't place a diagnosis, had no mechanism of a disease, and that was communicated back to the parents. However, in 2010, so we implemented whole exome sequencing in our routine diagnostic care. It had been there in the research before, but this was now into diagnostics. And we identified mutations in the gene called WARS2. I had no clue what WARS2 was, but it turned out to be a mitochondrial DNA-coded tryptophaneal tRNA synthase. So it's a synthase that actually loads tryptophan, one of the crucial amino acids, onto a tRNA, and then it migrates to the emerging protein synthesis and loads tryptophan into the protein that is being synthesized. And this enzyme is pretty crucial, and apparently in mitochondria, it's a very crucial protein. The mutations caused an instability of the protein, hence there was less charging. We identified through our network, besides these 2 boys, also 4 other children across the world. So it's a rare disease. And now we have 6 individuals with this kind of clinical phenotype, but also the same molecular defect. So this explains clinical phenotype, which is nice. But now we also had a prenatal genetic test. So this was communicated back to the parents early in 2011. And they had to make a very, very difficult decision. Should they go for another baby and have a prenatal test? What should you do with the result? So if there's a boy, high likelihood it would have the same defect. And if they would go into the hospitals in and out, the same kind of, they really didn't want to do that. For religious reasons, they didn't want to consider abortion as well. So that was a dilemma. If it would be a girl, it would be less a disease because these are maternally inherited. So in this case, they, after long thinking, they took the prenatal genetic test, and that's how they arrived in my lab. So what you can see here at the bottom is the father holding the thesis to which the PhD student figured out the mechanism of disease. And the mother was actually holding this lovely daughter of 2.5 who was just walking through the room, the lab, and just opening drawers. And they had a really good time, and they were shining all over. And they said later in one of our magazines from the hospital, thanks to you, our life got a positive twist. That's the Dutch version here. Now, the people you can see on the back here, all the technicians and the lab specialists working on mitochondrial medicine, the functional tests, genomics, that made this possible. And also the lab specialist, Professor Van der Heuvel, who put the puzzle together. And this is teamwork, and we all had tears in our eyes, and it's a very emotional moment, but this is the driver why we do this kind of diagnosis. And I want to share that with you because not only its impact in healthcare, but there's no therapy here. The diagnosis in rare diseases is half of the story. It's really a very important part so people don't blame themselves like they did. And the lesson here technically is that this technology innovation is really driving the impact in personal healthcare. We wouldn't have found it. It's crucial to combine different molecular views to understand human health and disease, which is now more and more into the multimodal diagnostics, which is not a trivial thing. And we need this fast translation of biomarker research to implementation, and there's a role of the academic medical centers. Now, we're here in a future of population health, and what I see since then is that next-gen sequencing definitely has taken the stage. First, in the diagnostic world, it was biochemistry first, proteins, metabolites, lipids. It's definitely into DNA first, and whole exome sequencing already for some years has become routine in the first pass diagnosis, very often a genetic diagnosis, and then you determine what other functional tests you have to do. The whole genome sequencing is being evaluated now, but it's quickly replacing these whole exome sequencing, but also many other genetic analysis platforms becoming the standard platform. And last year's in the Radboud Center, we evaluated whole genome sequencing, sequenced 100 individuals, and confirmed 97% of all the positive diagnoses already, but also could replace 10 different platforms into one workflow, which is for lab management much nicer. There is an exception. There's a few percent which we can tackle with a long read sequencing, and that's being geared up as well. Now, in the world, there's an increasing variant annotation in these genetic databases, more and more, and we just heard Professor Hood talking about all the genome databases and going beyond. In Europe, there's beyond 1 million genomes, so there's a large effort to do variant annotation. It comes from different associations, individual cases, as I just showed, but also by just new methods in the lab, but also in the analysis, and reanalyzing old exome data and more genome data. And the other trend which I see to go from population health is to move to biomarker panels, not just one golden biomarker. That's very much done, but it's more into biomarker panels. And that can be a combination of genomic, proteomic, a lipid, and a patient outcome. So we see more and more of that. So what we see now, and that's good to realize it's good to realize that omics has really a big power in these diagnostics. So the top panel, if you would analyze a single marker, you ask actually one question and you get one answer. For instance, biomarker X is going up a factor 2. If this would be a lipid and you would test all the lipids and you compare patient 1 and patient 2 in the so-called omics analysis, if this marker is still going up and the rest of the lipids is very much not changed, you get a very different clinical impression than when all the other lipids are being up and down, and it's a very disturbing picture. Cytokines, the same thing. The bottom panel would be a cytokine storm, the top panel will be a very specific cytokine response. So it helps to provide a higher diagnostic yield, give the example I just provided you, but also it puts the biomarker change into context. So more and more we do apply these omics into diagnostic care and an ISO certification, and it's really paying off. So by these different kind of platforms, clearly we can identify new disease mechanisms and translate to novel therapies. Now, it was mentioned a few times, but simple statistics don't cut it anymore. So we need deep learning, AI, and AI is really a buzzword, but we definitely need to scout what the application in omics as such is. There's only a few examples where AI and omics really makes a difference, but particularly in the link with other non-structured data like phenotype descriptions, clinical data, this will be very powerful. And I just showed you the first story on the diagnosis. The next story I want to share is on the diagnosis and on the therapy. And this relates to cancer. It's multiple myeloma, and it's the second most common hematological malignancy. And there's tumors that grow in the bone because there's monoclonal plasma cells that expand to a large extent. These monoclonal plasma cells secrete a protein, an IgG, which is called a monoclonal protein or M protein. Typically, the therapy of these are by chemotherapy or steroids, but also very specific, expensive drugs. Now, the specific thing about the multiple myeloma is that 50% of the patients ultimately get the disease back. So it's really important to monitor how much minimal residual disease they have. So that's the middle part, is MRD or minimal residual disease. At the end, at a certain moment, it relapses. So we need to start therapy as soon as possible when it occurs. And this has been a challenge because the diagnostic tests which are there, we test the M protein levels in the blood by gel electrophoresis. That's the standard test nowadays. So what you see if you profile immunoglobulins on a gel, normal, the gamma chains here are all kinds of sorts, but an M protein is very abundant, so you get a very distinct band. This is literally the diagnosis. Well, that could be better. But for mineral residual disease, those sensitivities are not good enough. So what happens is that frequently there's a bone marrow isolation. And then, for instance, these stromal cells are being analyzed by flow cytometry or genomics, take it a specific PCR or by next-gen sequencing. Now, the bone marrow aspirations is obviously a cumbersome, very invasive procedure. You cannot do that every day. And there is a very patchy tumor heterogeneity. So if you're lucky, you hit the spot. If you're unlucky, you hit a healthy spot, and then the diagnosis is false. So our dream was, can we use plasma and then protein analysis to monitor the mineral residual disease in this disease? Because the proteins that are being secreted is an IgG, as I just mentioned. And immunoglobulins have a constant region, which is just non-specific. It happens, it's a scaffold, if you like. But the variable region is very depending on the B cell that produces it. So it's patient-specific and multiple myeloma specific. This is like a barcode per patient. And this opens really the possibility to make a personalized diagnosis per patient. So the approach would be a direct measurement of this rearranged region by targeted proteomics. And the question is it specific enough? Is it sensitive enough? And we've used PRM, which is a targeted proteomics analysis, to do so. Now, first we tested whether there indeed are myeloma-specific clonotypic peptides, as we called it. We tested a large database with over 600 multiple myeloma patients, and indeed 100% had a unique peptide, and we can actually derive them, and they would be feasible for mass spec analysis. So personalized diagnosis could be possible. Then we actually took those gels, punched out where the spot is of the M protein, and applied this in the mass spec analysis, do the de novo sequencing if you didn't have genome information, and do a semi-quantitation. And indeed, in the panel, which you can see at the bottom, you can see that the orange line are the gel-based method, and at a certain moment we reach the limit of detection, but this M protein is still there. So if we apply the mass spec method, we can increase the sensitivity by 100 to 1,000 fold, and indeed can measure it much lower. If we start treatment, which are the yellow blocks, you can see that the M protein is going down, so the disease is indeed treated. But if you stop treatment, the disease relapses, as I just explained. And we can detect this very early on with the mass spec method, and at a certain moment, the gel-based method gets up as well. But by this time, the disease is already advanced pretty much. If you then restart in the same patient the therapy again, it oscillates down and it oscillates up again. So the principle works, this method seems to be feasible, and we can reanalyze thousands of archived gels to really do research here. But we want to get rid of the gel. So we developed a method to do this in plasma. And long story short, basically in a collaboration with several people who I just acknowledged in a moment, we could detect this mineral residual disease in plasma using a mass spec based test. So that's the MS-MRD, and we compared it with a next-gen sequencing on bone marrow aspirations. And indeed, similar sensitivity, it performed equally well as a prognostic biomarker. And if we separate the patients in plus or minus M protein, we can really have a good correlation whether it is the NGS or the mass spec. So that's really nice. And last year we published this, and it was selected as a top story by Clinical Chemistry, because this is where the whole field is going to. Clinical chemistry, with all respect, are still very traditional, old-fashioned, into one particular marker that needs to be measured in an absolute level. This is not an absolute level yet. It's as a change in time. So big changes, good trend, and this was very nice. So we took this further into with trials with IFM, which is a large hematological network in France, where we had many samples and we tested them at baseline, pre-maintenance, and post-maintenance, basically see the effect of the therapy. Again, the picture you can see on the top left is the limit of the gel-based method, and at the bottom was the limit of detection of the mass spec method, again between 100 and 1,000-fold more sensitive. And here we can really show in these samples with only 3 time points that if you would measure this by the mass spec method, we can detect early detection and early relapse, and the early relapse marker correlates with a poor prognosis of this trial, which was the patient-free survival, and that's the red curve you can see on the top left. So there's clinical impact here. What we're doing now is
1 minute to go, Alain.
Sure. What we do now is developing this assay into a diagnostic setting to make it feasible for ISO certification. Obviously, internal standards, QCs, we're moving away from the targeted measurements more into the data independent acquisition. For those into mass specs know what it means. And we improved this whole prediction, and this is ongoing. We clearly got some recognitions from the fields, clinical chemistry already mentioned, but also the medical immunologists. I have no clue what a mass spec is, but the principle here was really us noticed as pioneering MS work. So that's cool. Personalized cancer diagnostics. So this is only possible if you do this in a collaboration. So between the two university medical centers in Rotterdam and Nijmegen, we have a company called Sabia, who is really into multiple myeloma, several of the cardiovascular networks, but also two technology providers, Thermo Fisher and Bruker, who really promote this technology. Now, the lessons learned here is that we need to analyze those dynamic markers to restart therapy, adapt dosing, or change to another therapy. And particularly in the expensive medications, this is very, very important. Mass spec has a good value, good potential, and the collaboration is needed. If you scale that up to population health, then you can see that we need to scale up those profiling methods using blood samples, minimally invasive, but there we have a lot of work to do to standardize and harmonize. And what I said as the third one, to embrace the trend toward biomarker differences rather than an absolute level of a biomarker. And that's quite a big jump for the clinical chemistry. So to round it off, we have still things to do. Although it's nice in research, it's not good enough for clinical applications. So we need to improve the omics technologies. We need to increase the analytical robustness, fully implement it, the data fair at source. Fair was not mentioned, I believe, thus far. But very important, and we need to increase the capabilities to combine different data sets and particularly share and use the lessons learned, and I really like that. So if you want to know more about that, there's quite some papers about this, but it's not just telling it, but also doing it. So when we defined these principles of good biomarker practice since about 4 years ago, we implemented this in many different networks we're involved in. And I just want to highlight the X-omics initiative, which is a large roadmap initiative in the Netherlands, where we combine all the omics technologies into a combined X-omics infrastructure. And basically, if you want to learn more about it, LinkedIn is basically our community where we share a lot of news. We also do that on a European level as an EATRIS Plus project. So EATRIS is the infrastructure for translational medicine, and basically apply the lessons learned there as well. But this one is a pretty important one because we talk about technologies, but it's good to realize that we get different versions depending on the analysis we do, and there's no ground truth, and each of these kind of reflections will have a skewed version of yourself, and we need to decide how we deal with that for the own personalized healthcare. And with that, I'd like to thank you for the attention and thank a lot of people here. Thank you.